Welcome to Unit 3. Our focus will be on DNA and changes that occur in the nucleus that are related to the process of cell division. We'll pick up our class discussion on Chapter 10. Let's not forget what we've already learned so far from our previous units, and that is the tenets of life or cell theory. First, that all life is cellular-based. Second, life does not spontaneously pop into existence. It comes from pre-existing cells. So this tenet of life is reiterated here with a diagram of cellular reproduction where one cell, up here at the top, is the progenitor for all the other cells that come along after it, or below it in this case. These are called daughter cells, and they are identical clones of the original cell up at the top. The ability for cells to copy themselves allows for growth and healing after an injury. All clones or daughter cells are genetically identical, and the process of making an exact copy is where we want to focus our attention, and that process is called mitosis. Mitotic cell division is also known as asexual cellular reproduction, since no new DNA is being introduced. The term diploid has to do with the pairs of genes organisms have on their chromosomes. Haploid means half, and we will use that term a little more when we get to Chapter 11 and talk about sexual reproduction in the gametes. But for now, as we dig into Chapter 10, we will be looking at one diploid parent cell that duplicates its chromosomes and then divides to produce two diploid daughter cells. The process goes into a second round of cell division when we're talking about the sex cells. Body cells, also called somatic cells, undergo a single round of division, resulting in an exact replica of the parent cell type, which is the process by which your body will heal from an injury and is how you grow from a baby into your adult size. Asexual reproduction is the formation of a new individual by one parent cell without the introduction of any new DNA, so no egg or sperm. There's only one set of chromosomes being passed down to the daughter cells, and in this case, all of the offspring from this process are genetically identical to each other and to the parent. This process is known as binary fission, which translates as splitting in two, where an organism's single cell divides, creating two genetically identical offspring. Some examples of this are seen in both plants and animals, which of course mainly use sexual reproduction, but the eyes on a potato that you may have seen when it grows in your cupboard for too long, or the runners that you see extending out away from a strawberry plant, those would be examples of this type of process. Animal examples of this process would include a sea star, where they can regrow a leg, and members of the salamander family, they can also regenerate lost limbs and a tail. So we will apply this process first to bacteria and archaea that lack a nucleus and commonly undergo asexual reproduction called binary fission. They have a circular DNA structure called a plasmid, and the only way genetic diversity can come about in this case would be a mutation, where the DNA is altered, but it doesn't cause cell death. These phases of bacterial reproduction can be observed as first an elongation phase after the plasmid DNA has been copied, followed by a separation of the DNA into opposite ends of the bacterial cell, and then a new cell wall is grown in order to start dividing the cell into two separate entities. Okay, so now that we've introduced this process commonly used by mature cells, let's begin to apply the new vocabulary terms to what is happening in the DNA along the way. When we say chromosomes, what we mean is the DNA is packaged and condensed into chromosomes. Shown here is the chromosome pattern for a human. We have 22 autosomes that are numbered here from left to right, and a pair down here in the lower right-hand corner, those indicate gender. Those use letters instead of numbers. In this case, an XY indicates a male. There are specific stretches of DNA along the length of the chromosome that are called genes. Genes are specific regions of DNA that code for protein. Chromosomes are the condensed version of DNA. In order to get DNA packaged up and ready to move into the daughter cells, it has to be condensed, and this is done in an organized way. One long piece of DNA associated with proteins is called histone, which this kind of acts like a spool, like you would use for thread to keep it from getting tangled. So we can spool the DNA and wrap it around the histone, forming a nucleosome. And I have a big red circle around this structure. So we start off at the far right here at the bottom with our DNA, showing the double helical twisted conformation, and then we're going to wrap it around a protein called histone, which is in this orangey yellow color, and then the combination of DNA wrapped around a histone is collectively called a nucleosome. So notice in this diagram, the circled portion of nucleosomes, they're repeated, and then they get condensed as well. So in your ebook, you have a little different way of looking at this process. They've set it up in figure 10.4 as a table. So you've got your double-stranded DNA up at the top, and you can go to the next block down where it says DNA is wrapped around the histone. So you start to see that coiling process. And then the nucleosomes are coiled around even more in this middle box, and then further condensation of the chromosome to finally have this X-looking structure at the bottom. That's very characteristic for the packaging of DNA as it becomes condensed and ultimately packaged into a chromosome. They usually take on a little X-like structure. And we'll look at the anatomy of a chromosome here in just a second. Now, using a light microscope, we can start to identify those changes that we see in the nucleus. Let's compare the familiar-looking nucleus here on the left to the one on the right. They definitely look different from each other. When we see a dark staining, round, centrally located nucleus, that's usually what we expect. Most of the cell's lifetime is going to be spent in this state where you have the round nucleus visible. The DNA is loose and it's not yet compacted around the histones that form the nucleosome. But when the cell's getting ready to replicate itself, then we see the disappearance of the nucleus and the condensed chromosomes are freed up, available, floating around seemingly in the cytoplasm. Any dividing cell must first package its DNA, and these are now visible here on the right. It makes them portable, so we have portable chromosomes available to move around inside the parent cell and be available to go into the new daughter cells once the cycle is complete. So let's take a look at the anatomy of a chromosome. This is the DNA in its condensed form, and it looks like an X. This X-shaped structure with identical sides, and those sides are called sister chromatids. So there's a right and a left chromatid. They're held together in the middle by a tiny button-like structure called a centromere. The tips of the chromatids are called telomeres, and they are associated with the aging process. During cell division, the centromere splits, allowing the sister chromatids to move apart in opposite directions to become part of the daughter cells once they separate. Cells have a cycle that they follow, which is a set sequence of events that prevents mistakes from happening during cell division. Mitosis generates clones, and all DNA has to be replicated, packaged, and transported into the new daughter cells faithfully each and every time without mistakes. A cell's life cycle is divided up into two major phases, interphase and mitosis. We'll talk about interphase first. This is the butterscotch brown colored part of this wheel, and you can think of this as a clock going around. Interphase is when the cell performs its normal functions and duplicates its DNA for protein synthesis, and it spends about 90% of its time in this phase. The chromosomes remain in the loose, uncondensed state, and you would see the round nucleus, which is typical. And interphase has a G1, which is a gap phase, and the cell performs just normal growth and cell functions. And then there's a synthesis phase, or S phase, where the DNA is being replicated. And then the G2 phase is the second gap phase. It's roughly doubled in size, and the cytoplasm may swell, and it's preparing for cell division at this point. In the mitotic phase, it's highlighted here in blue. This is only a small slice of the pie, and even that is divided up into 4 smaller, more specific parts. In order, they are called prophase, metaphase, anaphase, and telophase, which is then followed by cytokinesis, and the 2 new daughter cells separate and go along their normal interphase business. An easy way to remember this order of events for mitosis is to use the acronym PMAT. P is the prep phase, and we'll look at what this means in just a second. M is for middle, because the chromosomes are all lined up in the middle near the equator when we look at cells in metaphase. A for anaphase means apart, and T for 2, meaning 2 new daughter cells. So if we go ahead and look at some normal cells, many times for practice we'll be using onion cells, which are what these are. They're stained, and it's pretty easy to determine what phase the cells are in. This onion root sample has several cells in mitosis. They have a red box around them. Clearly the nucleus doesn't look round or normal, but the majority of the cells here are in interphase, which is what we'd expect, since 90% of the time the cells are going to be in their normal growth phase. And you have a list here of the normal activities being carried out during this phase. Your learning goal for this chapter should be to associate the terms used in PMAT to what's going on in the cell. These images should help you do that. Stage 1 is prophase, so the P from PMAT, meaning prophase, is the prep. So we see here that the chromatin is condensed, and the nuclear envelope is starting to disintegrate. There's also a visible cloud-like formation on either side of where the chromosomes are, and those are the mitotic spindles beginning to form. They're made up of microtubules, and they form these little train tracks, if you will, that help guide the chromosome during the next steps. So M for metaphase would be stage 2, and the nucleus is completely gone now at this point. The chromosomes are aligned in the middle of the cell, kind of along the equator, and you can see that the mitotic spindles are at the north and south poles, and those spindle fibers are getting really long. They're reaching out towards the equator, towards the chromosomes. These proteins will attach to the centromeres, creating the kinetochores and microtubules that attach, and the chromosomes begin moving. All the chromosomes are aligned along the equator of the cell. This phase helps ensure that when the chromosomes separate, each new nucleus in the daughter cell will receive a single copy of each chromosome. In stage 3, anaphase, A is for apart, and you can definitely see the sister chromatids have been separated, and they're being pulled north and south, away from the equator. And in telophase, which is stage 4, T is for 2. This is where we begin to see 2 new nuclei being formed. The chromosomes have fully separated and are now located at opposite poles, and the spindle fibers retract, and the chromosomes are no longer visible under the light microscope. Early signs of cytokinesis are visible, and the actual microscopic image here, let me just go back for a second. Down here in the lower right is an actual microscopic image. The cell membrane has what's called a little furrow at the top, and this little V-shaped kind of a pinching in of the 2 cells is starting. That's also very indicative of telophase. You actually start to see two separate cells, and you can also see two new nuclei. So this will become a barrier and form a new plasma membrane, which will separate the two daughter cells. And we'll go ahead and we'll look at this next slide then. So in this one, we can see the cleavage furrow, a little more detail, and the separation into two new daughter cells is imminent. In an animal cell, the first sign of cytokinesis is the formation of the cleavage furrow, and then the cytoplasm splits. The term cytokinesis is the actual act of the two cells splitting apart from each other and marks the end of the mitotic phase. In plant cells, a new cell wall has to be formed prior to cell separation. So it's a little different here because you have a cell membrane and a cell wall when you're considering plant cells. So how this occurs is in phases. You see the 4 phases, reading this from left to right. So we've got anaphase to telophase. And what's happening is there are vesicles aligning, and they're filled with cell wall components. And they align and form a cell plate by fusing together and ultimately become the new cell wall separating the two daughter plant cells. You can refer to figure 10.8 in your book for more details on plant cell cytokinesis.